Hey, thank you so much. It's fun to be here. This is a topic, it's one of the topics I've thought the most about in my life, believe it or not. And so today I'm going to go through kind of like a mostly chronological story about how we've come to understand pathways of carbon in the ocean to the deep sea, largely through time series studies at a place called Station M. There's, of course, lots of other facets to the story, but I'm going to concentrate on that story today. And yeah, it's wonderful to be part of the Deep Ocean Observing Strategy and other related initiatives. This work is largely funded by NSF and the David and Lucille Packard Foundation, firstly through Scripps Oceanography and its affiliated programs, and then MBARI. And I worked at the National Oceanography Center in the UK for about 10 years of this work. I just wanted to thank OCB for their networking and events and workshops and so on. I've been to several of those, and they were really great to be able to go out and talk to folks about this kind of challenging topic. Okay, let's get these slides to move again. Here we go. Just a little more about where I am today, so that there's this global ocean observing system that has evolved since this work began, and I contribute to work on the biology and ecosystems themes, and I'm based at the Central Northern California Ocean Observing System, circled there on the right as part of the Integrated Ocean Observing System, and we're trying to build in more biology that can contribute to helping tell the story that we want to tell here, but it's still evolving. Okay, so going into the Wayback Time Machine to the mid to late 90s at a place called Porcupine Abyssal Plain. There's data there on the left side of your screen. And in that green shaded time period, there was some changes in the large animals quantified at the seafloor at 5,000 meters depth. Some really big changes that were not really expected based on what we knew prior. And so the changes were like orders of magnitude in the abundance of different fauna, and then some declines in other fauna. And what was going on, and was it just like measurement error, or like who knows? It was at the time it was really kind of surprising. It was data collected through net tows, so those have all kinds of challenges. And so when I was just starting out at the Scripps Institution of Oceanography working on Station M, we were looking at data of large animals at the seafloor through camera work, and we started to see similar patterns, right? So between right around the 98, 99 La Nina, El Nino, there was a really major shift in the fauna there too. Same kind of thing, like an animal per meter square, roughly at one point, and then this animal is like virtually absent some years later. And so what was going on there? It was a big question at the time. And so the tools that we used to look at this challenge included sediment traps. And so again, Station M is a time series station that nowadays it's a bit like the long-term ecological research stations that are now around the world. And it has sediment traps, and these are essentially rain gauges for particulate organic carbon and sinking particulates. And they can measure, they take a time series of these sinking particulates. There's also a time-lapse camera system at the site that can photograph the seafloor and sea animals and their activity over time, and then also photograph detrital aggregates that arrive at the seabed. So big visible chunks of phytodetritus, and you can quantify the aerial cover of that phytodetritus. There's also a benthic rover at the station that had been quantifying sediment community oxygen consumption. So this is like a food demand kind of term, like how much respiration is going on. And of course, the POC flux term, the sinking material, is a food supply term. And so this is like really fundamental ecology. And then we can also had been doing photographic and other assessments of what was at the seabed, as well as grabs, so like physical samples of mud from the seabed. And so this station had been operating since 1989. So that's the year the Simpsons started, and about 4,000 meters depth. And the station importantly has a lot of variation, as you'll see, and this variation can be kind of a real gift in terms of providing signal for trying to understand change over time. And so this figure is now many years old, but it still largely tells the story of connections between upper ocean variation and climate. So the topmost panel is an index related to the El Nino Southern Oscillation. Here it's called the Northern Oscillation Index, but it's a climate signal. And next, going down those panels on the right side, we have upwelling. So upwelling is a fundamental ecosystem structuring feature on the west coast of the United States in the California Current that influences primary production, export flux of that production from the surface ocean into the ocean's interior. And then we have these, again, these particulate organic carbon flux sediment traps as we go down. And there's two traps relatively close to each other nearer the seabed than the surface. And then the bottommost panel showing detrital aggregate cover. And we can see through these red and blue bars that kind of like tilt a little bit that these connections are frequent, like the instances of these kinds of connections statistically are kind of frequent. And the same kind of thing still holds today, just with a little bit more variation how that works out. And so we're kind of have got, I think part of the, what I hope gets discussed is how to move beyond these kinds of correlations. They're kind of reached their utility, I think. And so it should be exciting to dig into more thoughts about how to move beyond that, but a little bit more about the work that underpins this. So that I mentioned large animals, so smaller animals also part of the story. And the thing to focus here on is the topmost panel, where on the x-axis you have particular organic carbon flux, and on the left side axis you have total density, and then on the right side you have total biomass. And here the total biomass is correlated with POC flux with a response time of only a few months. And so that just is sort of conventional ecological thinking in a sense, and it was one of the first times we were able to quantify it over such long time periods. And although the scatter there is, the explained variation is not large, it's as best we has been shown to my knowledge. And so again, we're kind of reaching the limits of what's possible there. Here's another example of that same data type of data. So that this is data going again from like the surface ocean productivity, going down with POC flux as we go down the center panels here, and then sediment community oxygen consumption. So this is like, POC flux being food supply, sediment community oxygen consumption, food demand. And at the bottom, we have the macrofauna abundances in terms of density and biomass. And on the right side, you can see a relationship between density of these small fauna and the amount of food coming in from the surface and then to near the seabed. And this is only over basically an annual cycle, but but this is very different from the California Current high productivity upwelling area. Now this is in this case offshore Hawaii, oligotrophic, and a much more seasonally structured kind of a signal. And it's only 1 year where we were able to show this, but it was pretty clear what's going on. Okay, so started to get some better pictures about these connections, but still lots of questions about how they work and the details and so on. And so in the early 2010s, we started to see some more unusual changes, right, in something that we were not expecting. So if you look at the center and we go down to the third panel, there's zooplankton data there, and you'll see some around 2011, 2012, and 2013, some data that starts to pop out, right? Some really high abundances of zooplankton at the surface as measured through the Cal Coffee program. And then if you look at the third panel from the bottom, panel E, you'll see POC flux there, and you'll see some fluxes that are higher than any other part of the time series. And then next panel down with the red, you'll see self-aggregate cover. And what that means is the time-lapse photography at the seabed was seeing the seafloor almost entirely covered with self carcasses. And then the bottom panel shows the sediment community oxygen consumption in relation to that. So that this data is just as you see it there is really detailed in many ways. But if we go to the right side, we can see over that period of 2011-12 that there's this really high resolution data. So like in many cases, it's almost daily. So that the sediment community oxygen consumption measurements, for example, are on a 2-day scale. The sediment trap data on panel C is 10-day resolution. And we can start to see in real detail like how the food supply and the zooplankton and so on at the surface relates to these changes at deeper depths. But still we have the challenge of like, how do you make sense out of these things when sometimes they don't align and sometimes they don't quite align in the way you would expect. So it still leaves us with a little bit of a challenge. And here's another digestion of that kind of information on the left side. We've added to the kind of data that you've already seen. We just added mean and then standard deviation lines. The dotted lines are the 2 standard deviations. And then the reasoning there was to try and define what these pulses are. Like, okay, let's have a definition, and let's count them and measure them in a bit more of like a structured way than we had before. And that's what you see on the right side, is kind of revisiting how to visualize and quantify the links between, as you go down from top to bottom, again, export flux from the surface ocean as estimated by satellite, the trap estimated data, detrital aggregate cover at the seabed, and then the food demand, the oxygen consumption at the bottom. And you can again see these links, but they're kind of like inconsistent about the timescales and the correlations that are associated with them. So it still leaves us in a little bit of a jam in terms of having a mechanistic kind of understanding that beyond what we kind of have so far in this story. And then this figure, the thing to focus on here is really the bottom panel. And there's 2 lines there. You see, one is the sediment trap data running from 2011 through about 2019 or so. And you'll see that the sediment trap data is shown there in the black line. And then using what we knew so far, we took the estimated export flux data and then applied a Martin curve to it. And if you know the Martin curve, it has this factor in it, parameter called B. And B is it dictates like how quickly the organic carbon will be attenuated as it sinks, like how quickly it will be remineralized. And that's in part temperature dependent. We think there's a paper by Marseille et al. At PNAS in 2015, I believe, that outlined this temperature dependence. And so we applied that here, and we get this dotted line that is in many ways remarkable because it's almost exactly right in terms of the background flux. So it's like within a milligram per meter square per day on the background flux, but it misses the intensity of the pulses. And so when you miss the pulses, you're missing a large chunk of the overall carbon transport term. And so this remains, to my understanding, the big challenge, right? So like, how do we better quantify what's in these and understand these pulses? So, and since that time, we've tried to look at sinking particles, right? So in models, you can create theoretical particles and you can track them in the model outputs. And so one of the challenges, though, like you can ask the question, where are the particles in the trap that the trap is getting? Where are they coming from? So you can reverse the clock on a model and you can say, where did the particles come from over time? But you have to know the sinking speed in the model to be able to do this. And so if you've been following the sinking speed sort of debates, there's questions about the importance of sinking speed, but we can assert a sinking speed and then see where in the world and when the particles might have come from. And you can see there's 2 instances here, 50 and 100 meters a day. And those are annual plots there. And you can see it varies year to year, the range, but it can be from like Mexico all the way to San Francisco some years and some years not. And of course, it does have this sinking speed dependence. And so this starts to build a picture of what's possible, but still leaves us, it didn't, when we did all kinds of calculations to say, like sort of as fancy as we could get, if this improved our correlations in terms of like the connection between surface and deep ocean, and it didn't really do a better job. I mean, the correlations were sometimes better and sometimes not. And so it really wasn't satisfying to meet that need. And then in relation to that, we did an experiment, and I think this is one of most interesting things that's come up just recently is that in the model, you can say all the particles are the same, meaning like they have the same amount of organic carbon. So like if we release particles in the model everywhere the same every day, what does that look like? And so we have a trap in the model at the 3,400 meters depth, which is just a bit over the seafloor. This time series that you see there, it's a 2018 there, and the variation that you see in that, including with the pulses and everything, is only because of physics, right? So that the particles, as they sink, are getting aggregated into these ribbons. You can see the ribbons there a little bit in the animation. And this is just due to physics, right? There's no biology in this. And so we're still unpacking this, but you can ask the question, how much of the pulses might be due to physics versus biology? And so we're still, again, unpacking this. But it's an interesting avenue, I think, to conceptualize this sort of feature that we've been seeing. Right. So changing gears a little bit and to things that I think will be discussed at the meeting next week. So a few years ago, Boyd and others had this nice paper that outlined a little bit more clearly different kinds of biological carbon pump mechanisms. And this is really important to have better common language about how to define them and structure them in hypothetical situations so that we can do a better job isolating them in terms of their importance. Because what's happened in the past is that there are a laundry list of things that people assert as being important for the carbon pump functioning and so on, but very little chance of disentangling them in the experiments that were being proposed, a few years ago. But now it's getting better, I think, with this more common language and thinking. Mike Stuckel and his colleagues have had some great assessments of a sinking drifter trap, trapping sinking particles through drifting traps, which help eliminate one of the biases that we have known for a long time. And so they've got this nice array of samples producing profiles where you can diagnose carbon transport terms like Martin's B, and then starting to differentiate between sinking, active transport, and mixing, and having estimates that are associated with those. So this is, I hope, gets some attention at the meeting, and I understand Mike will be presenting. Also, so we have these tools. There's new technologies coming online with cameras and autonomous systems that are very promising. We have model frameworks, including like the Marseille example. They are a little bit unsatisfying in that, like, what happens when there's no curve fit at all, meaning like there's a big lump of organic carbon at depth and the Mark curve just kind of falls apart. Is a ongoing issue, I think. And then there are these climate models or biogeochemical ecosystem models that can run in climate forecasting models and other formats of modeling. There's an example from more than a decade now. But Monique Messi, Mike Stuckel, and many more are working on new evolutions of these kinds of things that should get some discussion. There's new cameras, and so just right now, while we're talking, there's this camera, this Planck de Boer camera that's known now, is out making an ocean section of biology down to 300 meters and about 30 kilometers long. So this will be a really remarkable biological data set, and it has 2 magnifications and delivers all kinds of information that can be used to help understand dynamics of carbon transport. And there's a deeper rated vehicle in preparation. So within 2 years, I hope to see this kind of data available down to 1,500 meters depth. Here's an example of an ocean section from a camera called Iris. So this one is a little bit more focused on rain snow than the previous one. And you can see this ocean section of biomass. And you can, from this, you can do things like calculate iterations of like Martin's B or other kinds of hypothetical constructs. The long-range autonomous vehicles, as I mentioned, are kind of a fundamental thing here. There's one available through the National Oceanography Center, Bodie McBoatface, as well as the vehicles here at MBARI, and there's no doubt others out there in the world coming online. And you can use these, again, to like revisit the Boyd et al. Mechanisms or other things. And so in the future, I would like to try and run ocean sections, repeated ocean sections, where you could start to isolate multiple transport hypotheses and estimations. getting near the end here, so that the sort of ton of image data that comes along with these methods needs a processing mechanism. And so Ocean Vision AI is a way that we're looking at a toolkit to process ocean image data. So FathomNet is a kind of an image annotation library and model library. The portal is kind of a practitioner space and processing space. And there's also a game associated with that for community science and engagement that can feed back into, can have community annotated data. And of course, the dream is to have visual data going in and annotated data going out, whether it's water column or seafloor. It works for both. So Leslie mentioned Synchro. Just very quickly, it's an ocean tech testbed, and it's focused on the Monterey Bay area in terms of its implementation, but it's global. It's open to anybody to come and test. We have 3 pieces of it, one of which is an offshore wind pilot study. And I'll just mention this because there's many new marine protected areas coming online and ocean wind farms. And so that we have a real challenge to try and understand them. But part of that for us is like doing exercises like what's going on today to try and understand how these vehicles and things can come together. It's supported in part by the fancy, like amazing modeling work happening these days. Here's an example of quantifying upwelling. And we can have this with a 3-day forecast now. And then we can send our fleet of robots out. And this is from this morning. So there are 2 long-range vehicles out, 2 gliders, 2 ships, and a wave glider. And they're all trying to look at biology in an ocean section along that line that you see there. And that kind of data can speak in the future as it gets to deeper sections, can speak to the challenges at hand. And so conclusions, right? So the time series are challenging to maintain and resource, but deliver incredible value. We plan to reach the end, I think, with some of the basics, and need to get into diagnosing how these pulses develop in a way that can be used in modeling. And the next point is to really encourage methods that are able to jointly evaluate a range of hypotheses, not just look at one. And autonomous systems are part of being able to do that. And that both there are size and type distributions of objects, large and small. So these systems can produce information that is really kind of overwhelming and goes across from like the phytoplankton, smallest phytoplankton, through to even large zooplankton. And if you looked at this, you'll know that you've heard an example probably of all of them being important. So you have to really capture all of them. And that bottom figure is an example of how data across these different technologies can really span a large range of body sizes and object sizes. And you can use that information in modeling is sort of the last point there. And if you don't have a modeling background, I don't, you can talk to somebody. And this is Deuce, the Deep Ocean Observing Strategy is a great community. OCD is another community with modeling and in-situ observers working together. So last slide, just lots and lots of people. It's hard. I was thinking about some of these names I haven't seen in a long time. Tons of people make this work possible. It's the list is not even close to being long enough. But yeah, so thanks.
So for those of you who joined us a few minutes late, our second talk is Amy Maas. She's at the Bermuda Institute for Ocean Sciences, and she's going to be talking to us about her work on the Exports Project. So some more big thinking happening. All right, Amy, take it away.
Thanks. And oh my gosh, Henry, thank you for all of the introductions. Like there's so many paired themes between what the Exports Project was trying to get at and what the long-term time series are trying to do. And so my focus today is to talk about more of like the biological, biogeochemical pathways and lessons from the Exports field campaigns. And to start with, right, the Exports Project, I am presenting work that comes from so many amazing people. And this is really a synthesis thing. And the general idea of the Exports Project, if you haven't heard about it, is like, what can we say about carbon flux from space? And so like, we know that we need a better like overall view of what's happening at a global scale, and that satellites provide that view, but that they're only providing like a surface view. And so to really get into that, we needed to put a whole bunch of measurements happening all at the same time to try to understand the movement of carbon from the surface to depth. And so experts supported this. So NASA put in this project. We had two field seasons. One was in the fall in 2018 in the North Pacific at Station P. So we were there during sort of a iron limited, low bloom, low export season. And then we had a second event at the Porcupine Abyssal Plain. So Henry showed some data from the deep ocean there, because there's long-term series, particularly looking at traps and the benthos. We were focusing on the surface ocean, and this was in the middle of the spring bloom in May 2021. And by goodness, it was a spring bloom. There was a lot of stuff coming out of the surface water. And so what I wanted to sort of do is kind of contextualize all this from a very zooplankton-y perspective of what is happening in the exports campaign. And again, the idea really was to try to see what's happening at the surface of the ocean, but then follow that Martin curve through the water. Who in the surface community is responsible for the export? Who is responsible in the midwater for attenuating that export? We are all familiar with the biological pump, right? So there's these three different kinds of pathways. We have this physical transport. There's engagements between bodies of water moving up and down. There's surface turbulent mixing, but also mesoscale features that allow carbon to follow pycnoclines. There's the particulate organic pump. So you've got phytoplankton producing the biological carbon. It's getting grazed. Everything is aggregating together, sinking out of this euphotic zone. And finally, you've got the zooplankton doing this migrant pump, diavertical migration. And so these are those words that Henry said that we're all sort of coming together and all using. Sometimes this is the ejection pump, particle pump, migrant pump. Those are the three things I'm going to be looking at. And in exports, we threw every technology that we could possibly think of and more into the water at the same time. So we had satellite observations, we had shipboard observations of light and attenuation. We had a ton of imaging systems, like the ones that Henry was talking about. We had towed systems. We had a bunch of particle traps, gliders for continuous observation. And the piece that I mostly focused on is that we had process studies going on. So we had one ship that was moving around trying to characterize the whole space that we were in. And then we had one ship that was trying to stay in both conditions in a retentive system. So in the North Pacific, that was easy. Things weren't very dynamic. In the North Atlantic, we were in the center of a mesoscale feature trying to stick with a floating trap, a sediment trap, so we could see what was happening over time. And the structure of the design was that we were there for 3 7-day epochs, and we were trying to see the change in flux over the time period. And so this is the context. And although, you know, this is a conversation about what's happening in the deep sea, as we all know, the source carbon primarily is coming from the euphotic zone, but then is moving into the twilight zone. So when I'm talking about these things, yes, the exports program was all like surface, but like the cool stuff is what happens in the midwater, says the zooplankton person, of course. All right, so in Amy's perspective, exports was about understanding the relative contribution of animals to midwater ecosystems. And I'm breaking this into supply and demand, right? So we have the supply of carbon coming from the surface. And when I talk about these things, then I'm looking at the relative animal contributions. And they're really big players in 2 of those major systems, right? Both in the passive particle flux, and then the active migration of carbon to depth. But they also play a role in this demand, because if carbon just like fell through, then we wouldn't have that attenuation. We wouldn't have that Martin curve. Of course, microbes also contribute to that attenuation, protist or bacterial, but I'm interested in understanding how the animal component is affecting that attenuation, the distribution and the physiology of these organisms are changing those things, and also what that means for midwater communities, right? When we think about the animals, I tend to break them up. We've got a surface community, we've got a mesopelagic community, and then we've got migrators connecting in between. And they all kind of have different physiologies, which I don't know if I'll have time to touch on, but that's where I go crazy and get excited. I am an organismal physiologist who is now dabbling with modelers. So to give you context. So the zooplankton group, who were we? So Deb Steinberg and I were co-PIs on the Zoom group. We had this wonderful postdoc, Karen Stemieskin, and some phenomenal technical staff who came out to sea with us on these cruises. And what we were doing is we were taking this 1 meter squared mock nest net system, which trawls in different buckets and lets you see what organisms are at different depths. We had 200 micron mesh nets. These were depth stratified. We're going 0 to 1,000 meters. And what we were looking at is the biomass of dry weight, but then these image analyses. So ZooScan analysis, this gives you abundance taxonomy and that size spectra. That size spectra is what was shown in the last slides of Henry's talk, of trying to understand how this matters. And the reason I'm really interested in that size spectra piece is that I do rate experiments, and rate experiments on large organisms relate to their size. So we call this allometric relationship. So smaller critters breathe more per unit mass than larger critters. And so I was doing measurements of dissolved inorganic carbon, so respiratory carbon, dissolved organic carbon, and then our postdoc and Deb were focusing a lot on the particulate organic carbon fecal pellets. So these were our methods. But I'm not really going to delve into the details of what our particular findings were because, as I said, experts is this phenomenal collaboration. And I think it really highlights how important it is when you get everybody in the room with all the tools, all talking together, all working to try to understand each other's terminology to say something more. So what I'm going to show you today is actually the product of all of our collaborative work. So these are just the PIs from the Exports project. The whole project was more than 100 people who spent a great deal of time pulling together data sets. And so the information I want to show you today is mostly synthesis projects that we've been pushing forward. So again, starting with Station P, this was the 2018 one. We're a lot further along. One caveat about the, yay, it's amazing to work with everybody. It takes a lot of time to work interdisciplinarily and to build synthesis products and to make sure that everybody's units are in the same units and that the way that we measure, like with a net toe versus a CTD bottle sample, is cross-comparable. And so I'm mostly going to be able to give you a polished talk about the 2018 data just because it takes that long for us to figure out what we were saying and what we were doing. So there's that piece. Now, as I said, I know we're focused on the midwater, but our first step is always determining the supply term. So we're trying to understand what's happening in the epipelagic first. So this figure represents a carbon-based food web. It's visualized as a set of biological carbon stacks. Those are the boxes, right? So I was responsible. Oh, can I draw? I cannot draw. I can't. I figure it out. I will not figure it out. Laser pointer. Nope. Can't do it. All right, the screen. Mesozooplankton. This is us. Mesozooplankton. Salps. Salps were really, really important in the North Pacific. As Henry said, when they are there, they matter. And this was very clear during our data set. These stocks then are connected by rates, and these are these filled in arrows that we're showing throughout this figure of carbon transformation. Now, all these values represent cruise averages, and most of all of these were directly measured. Okay, like all of the stuff that I'm showing here, like that the North Pacific is a generally recycled community in the fall, is not like new news, but the fact that we've measured all of these things at the same time was incredibly important for us because it reduced the uncertainty, which was one of the major goals of the Exports project for modeling purposes. So there were some rates that were calculated, such as like assimilation efficiencies, which are from the literature, and there's asterisks where those are. And then the other kind of part is that the zooplankton were captured from 0 to 1,000 meters when we measured their rates, but then when we compare with literature, we can say what that means. So this product basically says, in this system, carbon is recycled. Okay, this is not new. However, this low productivity general stable recycling system, we could actually demonstrate that there was really high grazing rates by the protists. We were able to measure that. We had isotopic measurements showing that there's at least one protistin trophic step in the food web for all zooplankton size classes. And then the only really dynamic thing was these salps, which would come and disappear and go away again. And since we were able to measure all of these different pathways, the predation rates, the grazing rates, the loss processes, we were able to sort of attribute what was coming out of this surface component and to divvy up amongst the different components. So microzooplankton had a tiny role. There was very little aggregation or detritus, and everything that was coming out was from the zooplankton. So you had a bunch of mesozooplankton flux coming out in the traps and a bunch of salp flux coming out. And that was particle. But one extra piece of that is not all zooplankton are the same. So as I said, we had very patchy salp distribution. And when we measured things like respiration rate, dissolved organic carbon, and particulate organic carbon or fecal pellets, the ratio between mesozooplankton, which were generally crustaceans, versus these gelatinous salps is very different. You'll notice this is the fecal pellet of this little copepod, and this is a honking fecal pellet of a salp, right? Like these guys are Wheaties. They are just pumping that carbon out. Sinking rate matters. This is going to sink a lot faster. And if we infer their grazing rate based on how much carbon was in the fecal pellets, the salps were doing a great deal more grazing than the mesozooplankton when they were present. So we have more fecal pellets produced by salps. And actually with the mesozooplankton, more of what they're losing as carbon proportionally is this respiration and mortality fact, right? And that is mortality due to predation, not due to like senescence or just like, I'm done with life, I'm gonna die. And so we have very different pictures between these two different types of organisms. As Henry said, tracking salps is really hard. They boom and they bust in figuring out where they are. And if you actually look, there's a salp paper that we did that showed that they flowed in and out of the dynamic system. One thing that we saw from that last slide is that, here, I'll show it real fast, zooplankton actually weren't meeting their metabolic needs with their grazing, right? There is more food coming in than going out in the salps, whereas that was not the case when we measured the respiration rate and the fecal pellet production rate of the zooplankters. And so if we add that all together for the full community, we have a net decrease in the carbon that's happening with the zooplankton. Now, this isn't particularly surprising. We are in the fall, low food season, and the zooplankton, we think, were actually unable to meet their metabolic demands versus grazing. There just wasn't enough stuff in the water for them to find, right? And if we think about what we know, spring blooms in this region, fall season, that zooplankton are in some ways the batteries of these systems, right? Like they consume a lot of carbon in the spring bloom, and then like over time, they're like, losing their carbon. Some of them die, some of them are lost, and they're just like eking away until somebody makes it back to the spring bloom again. So we think that there's this biomass accumulation that happened earlier in the year that is being lost by these zooplankton over time. And because they're metazoans, they can starve for a while, right? Like they can not consume something and just keep releasing carbon that is stored into their body mass over time. Now, if we try to look at that then for what this means for the supply term coming out, what we've learned from, again, from the zooplankton perspective, what we see, this is, again, the breakout where you have respiration, dissolved organic carbon, particulate organic carbon, and mortality segregated between the zooplankton and the salps. And this is what I mean by patchy. Sometimes the salps were present and they were producing substantial amounts of carbon via active flux. And sometimes they weren't present at all. But the mesozooplankton played a larger role in moving carbon through this active flux pathway. In contrast, this is data from the traps. Colleen Durkin, Megastapa, and Melissa Oman did a great job of visualizing the flux in the traps, and we're able to attribute salp fecal pellets as a subset of the total particulate organic carbon at all these depths. And so like when salps were there, my Lord, those poops were important, right? So this light blue is every time that we have the salp fecal pellets, and the dots here is the total POC. These are actually modeled based on what we were seeing UVP and the particulate organic carbon in the traps. And so sometimes salps were absent, we had lower flux, and sometimes they were present, we had substantially higher flux. So salps were really important for this passive flux pathway, which again, emphasizing exactly what Henry said, when they're there, they're large, they come to the bottom. And we didn't calculate what would happen after this bloom senesced and died and you get a jelly fall to the floor, because that wasn't what we were seeing yet. Their mortality term had not yet happened. They were still in a bloom phase. So this is what we expect from the supply term when we think about how things are coming through the water. And if we move into the mesopelagic, we had similar to that epipelagic budget group, there's another set of exports collaborators who set up to build a budget for the midwater. And so this data set's based on cruise period net community production, which was estimated from the gliders. So we had gliders that were put in before we went, and they represent the difference after subtracting particle flux measured at 500 meters, right? So this is what is moving through this 100 to 500 meter term. And we're just trying to figure out this box of the mesopelagic, this first area in the model. And so we get the supplies, and again, these are measured using multiple different methods. And we have a whole conversation about how the different methods inform and constrain our uncertainty on the different components of mesopelagic supply of carbon and mesopelagic demand. And so when we put this all in, we have the typical, everyone gets there, is that there is more demand in the mesopelagic than there is supply. Again, this is not new. We've seen this over and over and over again in the literature, but we've reduced our uncertainty. We know that all the things that it's not this, it's not this, it's not this, it's not these other hypotheses that we've said it could be. And so that lets us really dial in and say, okay, it's here, what's happening? And actually what we think is we might know where this is coming from. We don't have the smoking gun, but we think this might be supply from earlier in the season. The same sort of conversation I said about the zooplankton. We did this as a Monte Carlo analysis because we had so many different types of data coming in with slightly different values to give us some uncertainty. Our general estimate of what's going on is these are all our supply terms with all of the uncertainty constrained. These are our demand terms with all their uncertainty constrained, and the difference between them then is this excess demand. So the midwater in this region was 2.7 plus or minus. That's the amount that the mesopelagic is in excess of steady state. And remember, this is like a stable recycled community that was like nothing was changing. But even in that context, there's not balance. So I think that there's something about the word steady that makes us think balanced. And we've known this, that this isn't true, but it's not because we're doing a bad job of measuring. And so what we have for data that shows us some really interesting pieces is that during the cruise, Brandon Stevens, who was working in Craig Carlson's lab doing dissolved organic carbon measures, made profiles of the dissolved organic carbon going through the water column. So starting at 85 meters, going down to almost 500. And the blue dots are at the beginning of the cruise, and the red dots are at the end of the cruise. And this is a really precise method. This is beyond the level of uncertainty, but barely of the machine. And what we're seeing is that we have consistent dissolved organic carbon removal throughout the water column. So the microbes are pulling down this dissolved organic carbon and reducing it. So we have a stock of dissolved organic carbon that's in the water that is supporting microbial demand in the water over time. Similarly, we've shown that the zooplankton are potentially starving and are losing body mass, are not grazing. And so that was primarily done with surface organisms, but as they migrate down and as the mesopelagic organisms who have stored lipids lose food over time, that those pieces might actually be adding into the oxygen demand of the midwater, surviving on carbon that was fixed at a temporally and potentially spatially distinct location, which makes it a little tricky for modeling, but something to think about. So if those are the two big budgets that we're thinking about, and the big takeaway lessons, at least for me from that project, is that this sort of interdisciplinary work, yeah, it's hard. And even if you don't change the paradigm, what's really critical is that we are reducing the uncertainties of how food webs like this, which are really low signal to noise, how they work. And that's really important as we start to try to model this stuff going forward. Same thing Henry said, flux can be episodic and punctuated. And it's almost like you have this recycle system, and the only way that carbon can get out is if like a gear slips and suddenly something blooms really fast. And then it like, the carbon comes out as a massive fecal pellet. And so these are rare events, and those are also really difficult to capture. And even these stable systems are not steady state. Direct measurements of rates also is extremely useful, even if it's hard. And that's one of the things, like, I love autonomous things. I love all the instrumentation that's providing image systems that allow us monitoring, but we do still need these process studies because rates is something that's really important. And a lot of the things that we're catching are really ephemeral. Like if a zooplankton pees in the midwater, like it is immediately taken up, right? Like it's hard to measure that from measuring a Niskin bottle of what's the DOC, because the microbes are gathering it, and then when we filter it, we lose it. So it's patchy and it's temporally distinct. Okay, moving on to PAP. I cannot give you as like coherent full budgets, but I just wanted to show you where we're thinking and where we're going. The North Atlantic was a completely different place. It was profoundly stormy, and there are such cool interactions between physical aggregation and disaggregation, a very different idea of what's coming down through the water column. In the Pacific, it was all fecal pellets. Here, this is aggregate city, right? This is phytodetrital heaven. These are the traps over the duration of the cruise. This is looking from the surface to depth using UVP. And this is aggregate abundance in numbers per meter squared. And so the contours are what the UVP was counting, the number of aggregates in the water column. Every time we have a white spot, that's because we were stormed out so bad that like nothing was allowed in the water, because would not have been good. And each of the dots then is specific either traps where we're actually taking water samples of things. So by way of passive flux, this system was completely different. There was a ton of flux and it was primarily phytodetrital. Just to give you context on the active flux, this is like the average active flux from the North Pacific. This is the average active flux from the North Atlantic. And this has to do with the fact that there was just a lot more organisms and they had a very different size structure. And the active flux played a less substantial proportion of the role of the carbon that was being moved through the water column. Even our really big animals, like we had these huge pteropods, and you'd put them in a beaker with filtered seawater, and they would produce fecal pellets for like 24 hours, like insane amount of particulate organic carbon. But when you compared like how many of there were versus how many aggregates, it didn't even matter. Like it was a ton at one time, but there were just so many aggregates that it wasn't, yeah, pteropods. The other thing that we're trying to do in the North Atlantic is recognizing that fish really matter. So this is the best synthesis I have thus far of flux in the mesopelagic. So the supply of carbon, and this is to 200 meters. So this is bringing in, this is our thorium estimates, and this is ranges of uncertainties. This is from the first week, the second week, and the third week. These are from the traps, first week, second week, and third week. Remember, these are integrated over time. These are shorter time period. Oh Lord, time periods. Zooplankton started off a similar level of flux as the particulate organic carbon, but towards the end, the aggregates just dominated. And then we start to add fish, right? And although fish are substantially less here, being able to go up that extratrophic level is important because zooplankton don't migrate as deep and their fecal pellets are smaller, so they're less likely to escape even deeper into the water column. So when we start to think not only in the context of export below the 200 meter, when we start thinking about really sequestration or fecal pellets making it down into the deep mesopelagic, fish contributions are going to be a much bigger deal. And so we're starting to add that piece to the North Atlantic. Okay, I was like, oh man, what should I tell all these people? I'm this observationalist and this physiologist. Okay, so what do we need to do in my humble opinion? So we have some gaps still left in our observations, and the technology that we are starting to implement is just so cool. So like all of the continuous measurements, all of the imaging systems, all of the metabarcoding, like those are important, but we are still missing some really major stocks that we just don't have. Things like dissolved organic carbon, things like midwater zooplankton and fish abundance, because who wants to be dragging a net through the midwater for zooplankton and fish? It's hard. Like most of our observations of these organisms are from the surface. And they like run away and have behaviors. It's just a pain. We also do need real weight rates for midwater interactions in physiology, because that that changes systems, and they don't behave the same way as surface organisms. One thing that I harp on and I don't really know how to solve is diel-vertical migration. We actually never measure that. It's always calculated, and it's always based on biomass. And that's something that we don't have well included in the models, and we're getting there. There's a lot of interesting and new approaches to how to do that, but that's a problem I'm thinking a lot about. And then in models, there's complexity of midwater niches. And I don't just mean that for my metazoans, like understanding microbial interactions with particles, microbial interactions with dissolved organic carbon, recalcitrant versus non-recalcitrant, what things are doing in this realm, which historically we thought was super boring and devoid of life, but it seems to have such complexity in these niches. As Henry showed with the filaments of particles coming together, these things are aggregating and disaggregating even without the help of biology. And biology is absolutely going to throw a wrench in the plan and try to get even more complicated, because that's what it does. Particle size distribution is critical. We keep trying to add more boxes to our models, but like that just doesn't accept the allometric relationships that the physiology of the larger organisms at least really require. And thank you. Again, I didn't even try to put everyone's name on this slide because it's impossible. The experts group is just so good, and you should read all the papers and you should like all the people. And that's my story. I'm done. Thank you.
So you guys have done, you're talking about two different things where you've made some really, really big headway on these pathways, but one of them is a dynasty like The Simpsons, and the other one is everything everywhere all at once. And so it's those are like really unique unicorn situations, right? And so I think what we're going to be trying to do at this workshop is trying to figure out maybe those other little pieces. Maybe instead we're making, I don't know, Frozen #3, and like what is the cinematic thread that we're having? Like where can we get the biggest bang for our bucks, or prioritize recommendations, or maybe we do figure out the next cinematic blockbuster. But yeah, this is, I think all the advances that your groups have made have just been really great. But yeah, we have some time for questions. Anyone have any questions on the floor? Yes, Irina, go ahead.
Oh, yes. Hi. So I'm curious, a question coming from, global biochem modeling perspective. So know little about the exact details, but I lost when you were talking in the North Pacific, and you showed some interesting picture of the DOC and said that there's a lot of DOC removal throughout the column up to 500. And then you're trying to connect, maybe you can restate that. How are you connecting the zooplankton at the surface and the mesopelagic organisms and the DOC story? I lost you there somewhere a little bit.
I think they're two different processes that are separate at different trophic levels, right? So I think some of that dissolved organic carbon that was produced earlier in the season was mixed down and was produced in prior time periods when the consumption did not outpace the production, when you had less of a recycling in the surface, right? So that dissolved organic carbon was produced at a different season. I think the zooplankton as well, if I think about surface or midwater zooplankton biomass production, if you have a lot of carbon fixation in the spring, and you've got organisms consuming that carbon either in the surface or as aggregates fall to depth, they're putting that carbon into their body mass, right? And later in the season, when there's less carbon coming from the surface, either in the form of migrators or particles, they're burning that carbon that is part of their biomass and basically releasing carbon without, they're consuming their own bodies, basically. So it's a starvation piece. And I think eventually that results in them starving and dying because they're not meeting their metabolic demands. But in that case, they would move into the particulate field. So I don't think that there's necessarily a direct link between the DOC that's in the midwater and this idea of zooplankton slowly starving over the annual cycle. But I think it is both indicators that when we look temporally at a place and say, it's steady state, that's not accurate even in something that's very, very stable, because the whole system is evolved around a prior spring bloom organisms are expecting that is going to happen annually. Does that make sense?
So, but your DOC in the, deeper, I understand at the surface you have different types of phytoplankton and bacterial plankton that consume your DOC, but deeper down, is it being consumed? Also?
Yes. So that
It
DOC
continues to be consumed.
It continues to be consumed. So that was 85 meters to 500 meters. It's the microbial community in the midwater is continuing to consume this stock of DOC slowly over time.
Mm-hmm. Mm-hmm.
And so they're using up the oxygen. So that's part of the biological demand. And it's DOC that is not, it wasn't fixed proximally in time. That makes sense?
Yeah. Okay. Good. Interesting. Very interesting.
Kind of reminds me of like bears gaining weight in the fall, and then like just to put a terrestrial example on it, big tubby squirrels in the fall. Yeah,
Burn it down.
Yeah.
Yeah. I mean, there's also the lipid shunt happening. So during that time period, we also had the ontogenetic migrators coming down to depth, which is another one of those like stored previously coming down midwater food option.
But can you separate inside the DOC? What is the kind of
The different compounds?
the different sources of getting the DOC there? I mean, it's just how is it being produced and the different sources for it, I guess.
I don't think we have a tracer on it, particularly with that level of the rate of consumption. So we could look. It would be interesting to go over the annual cycle and look at the moieties of DOC, like the metabolites, right? So is it a metabolite that is coming from animal tissue, or is it a metabolite coming from phytoplankton tissue? There's certain markers we could use. And if you did that in the midwater over time, I think you could see ratios, those things changing. But again, this was a process study. This is why, Leslie, you can't do only one. You have to have time series and process studies, right?
So, and I guess the other question is, this association between the POC and DOC, right? And the ratios between them. It's very interesting.
Absolutely.
You guys are already writing the next proposal.
Yeah, there you go.
Oh my God.